Ooh. Shopedale? Excellent, good. So I hope you're all doing really well. I'm finally getting over my jet lag, although I'm still kind of struggling, but I'm doing a lot better.
Shopedale, ane chesu sama, ane karsata, debut hambu yo periwachigi yes, tanda imbana, ta tizu tude tanga se tinzu ngetu te chungo yorwa, ta tanda this that order shas, tanda imbana tata chita chomendus imbana tanda the ostus.
All right, so yesterday, Kelsey did a beautiful job on introducing and reviewing some of the things that we saw last year and new things that we're going to look at this year.
Kesa ge kesi lagi bayna ngasugu lungoma kare jang yo bata ane talu ngasugu sarba kare chi jang yo mete la ya ne ngotoros de pe ya puchna sons.
And we finished the day with our experiment, so don't forget to look at your results every day at the same time. Okay?
Ta ngasugu kesa takshe chi dribyo res wa ne takshe dribya te la ya nima tapar titsu chipech ke gang la ya ane kanda chi chondo te la ya zju ma jo na jes.
And what is really interesting is that our experiment is based on seed germination, plant growth and development, and for today we're going to look at the details of plant growth, photosynthesis, and really what drives the process of photosynthesis.
Ta tanda nga suge takshe shimji chete kare yore system balaya ane tampu telaya ane ko sabuni ni kumbiwa ta tene nga suge sarpar chongwa ge ramba te mampu chi yore swa ateta te nga suge takshe ge nimpu chi chagores ta tere imbana nga su kare tagi in system balaya ni kumbiwa ta ni kumbiwa te ki jese imbana ta ko na ne Ojo chia chuarwa, luma gana lai ojo chi de te kogi nubati kanran ru tungu yo me tigi thalaya nga suge should support on the
So the three things we're going to focus on for today are going to, of course, be photosynthesis and the driving molecules that allow for the process of photosynthesis. And for that, we need to understand the chemistry of water and atomic movement.
Ta singa sugu ojo ko la tagres ro ta ugi debjurti kanadu yo met singa nane ro ugi debjurti tagres ta ugi debjurti taya la ya ojurti temperature gi dudu karim bate tagu yo res dudu karim bate tagu yo re te tabi kapsi bana ngasu kar tagu res na chugi dudu la tagu yo res chugi dudu la tabi kapsi bana ngasu gi chutriya gi dutrengi chechu ji ro te tala tagu yo sanga sugu dutren te yan discuss ngosu chi gi yesku chechu zu zong de.
And for the afternoon, for the third session, we're going to do two different experiments where we can actually visualize the process of photosynthesis.
Ta ningon jala imbana nga sugi takshe shimji dribi kapsi chego ni chi dribi yes chi kare system balaya ojur chebi kapsi rimba kandan ro chin ro yo sate ta visualize sig do ta ko yamna samla tra yamna remote do tria do chim do odena do chi ta chi thombi gan sigura samdo na chebi ngu gane ojur sa gi jirimti kanda yo mete ta yes.
All right, so let's begin. These are our objectives for today. Things that we need to be able to understand. We're going to work on them together and we're going to focus on them throughout the day.
Ta tanda de tampo nga sugal hotriga mugiti yes lotre mugiti kasna nga suge kari chi siala riwa chi yo patang kangi gala ya shu jagu yo meta yes.
We want to understand the properties of water. What allows water to be such an important molecule for life.
Ta chugi chechu tingunzi chi yes chugi chechu ngunzi chi be nane so de chu sete tserolai rwa tserolai karchine ke chambu yeme ge neta de tagis.
We're going to talk and explain plant development, essentially the steps of plant growth. How does a plant go from germination all the way to adulthood when it reproduces?
Ta nyotela nga su tsekhang ge tsapre la tagi tsekhang tsapre tabi kapse ko tangpo nga su tsekshin chunchun ne da ko ka sabune yang khore dan bi yong goro chonchon ne nyukum biwane song de te ani ko la ya tsapre jurum kanda chikdu thamatele ko ge yang tshan te ge chepe chia ge ramba chile garwa o de ta ramba de kanda yeme te tagins.
We're going to talk about photosynthesis.
Ti chila nga su ge ojur kolende se jagins.
Now, I will say, this is the same topic, the same talk that I give to my students at Emory. And photosynthesis is a subject that they find very difficult.
Aha. Ta dandagi ngi chetsula ya ojur ko la thiagi lhamsa tagaran jangi emery gi lobtu chula yan trigi yes kusu imba na ojur gi zinda de tripikaps zinda de lhamsen de hachan khapu chila yongin jigres.
So what we're going to do as a group is we are not going to worry about all the little details of photosynthesis, but we're going to talk about essentially the main steps, the main points of photosynthesis on a bigger scale to make it simpler, allowing us to understand it.
Aha. Tanga suge ojur ko la ya zinda thiba ta di jela ya jego tripikaps imba na nga suge ship chapi gi chashi re la ya nga su dus tag mes. Tige nani gomba ta concept gnani ke chishu kange resha titong win zamba titak chelit zungu shimba che chi chane imba na chiyongi ojur sa ti kanda do chirin do sa ting win zamba che la ya thapchi chigins.
And like I said before, the afternoon session, the third session, we're going to do 2 different labs, which really allows us to not only understand photosynthesis, but to see it happening real time.
Aha. Ta yingkong jabla ya nga suge chegu drip pickups chikari chigin sena ojur sati kanda do chi chungi yome tige netante nga suge kasugura ta takshe chegu gangne dribate chi cherwa tige gangla ka system bala ya ojur di ani tongis kandas chung du sati nga suge ta muthon laksan gangne ge ta nyams nyoya thoya chi ge thapchi chigis.
And it's very exciting because these are the labs that I teach our students at Emory. So you all get to do the same things that Emory students do back in the United States.
Aha, ta tanda nga su nyingkong la dribya ge taksi chegu de imbaina this tangwan demba tinda yores dua tang demba tinda yores tanda nga su ge nyingkong jabla che ge chegu de tagaranja Emory la ya ngaran zinda tripi kaps lotu sula taksi tagana dribki chigi yes.
In the first experiment, in the third session, we're going to measure stomata. These are the little sort of organelles that allow for the gas exchange for photosynthesis and for the exchange of nutrients for a plant.
Aha, ta chikar tagre system bala nyingkong asuge longkong sa sto longkong sa de luma go gangla yopa ga ta session go luma gangla yope ge ko ge organelle ta wang tha ching ba kan shila ku kar chigre sena kasa de langzi chigoro langzi de zo chilo ta na lo la dreng chiga ta shenda yang ku ge si gi chu de yang chilo na lo dreng ba chia la chigres nga su ge nyingkong jab la ya longkong gi tukse de kan do chirin do te tagis.
For the second experiment, we're going to visualize the exchange of gas and how leaves change their density, and that is a really, really intricate and beautiful experiment.
Aha. Ta tigi jela nyoti kare system bala ya ngasuge ujur seti tongis kandan dogun jungu yoba tela ya samutang de ta tela tene de te ani luma ge tsela jiran do achiga ro otela ta ge instati imbana chetang ge da ko takshe chegu na ne takshe chegu ta yan sambo ngoni yapuji yores.
So here is your homework before the third session after lunch. Before you come in for the last session, you're all going to you're going to go out, you're going to collect leaves from different trees, different species, and bring them to the class.
Aha. Ta chesula ya nangli chunchun chi yes te kare sena nyingkong shela ge je ge choton te ma chungo la ya ni chesu chama ge wan zanda som thote chama cholo la phe de te ani shindong gang ge loma rig mando wa ani dus duruchi yong gu res.
I would try to bring about 20 or 25 leaves from different types of plant species. Okay.
So 25 from each group or for each individual?
Leaves, maybe 3, 4, 5 different species, and overall about 20, 25 leaves.
Okay, okay. Yeah. For like whole class?
For so everybody gets 25 leaves, so that way they can try the protocol. So everybody collects 25 leaves.
Okay, okay, okay.
All of you collect 25 leaves.
That means re ge imbai na roa. Mi re ngone luma mandawa nistanga sidigu yo resra lunga mandawa tati imbana luma mandawa tinda ta karsa nistanga chi yong ba bana tila tene deta ya seshingi nyirig mandawa ta peche shinga chi yong res tati sama susuk ta yes.
So on your notes as you're collecting, make sure that you note what species the plant came from, so when you come in and you get the data, you can link your data with the plant species.
Ta luma de dere chipe kapsa luma de tshinga nyiri kari ni to yente 수수기 드템나들이 퉁고지로 제스와 퉁고로 제스 네 되게 제라야 아니 차통 치셰 올란두 스타비 값스 아니 로마티 거니 오니 하고 그레스
And make sure the leaves are fairly large, not damaged, or not yellow or red. They have to be fairly green and healthy.
Danda Luma Dibicups you said fairly large.
About maybe that size.
Okay. Kala Kansan Chamberwa, Kala Kansan Chambuchi, and ko Luma imbana Tambu Sagurata, Janku imbarwa, Chi Serbu imba, Chabu Chabadota, Yana Kotola, Tapando, Yerdo, the Maimba, Neko Tambu Yoba Sagurata, Yabu Yopatenda, Terrachis.
We're good?
This is
Excellent. Kelsey, anything else? We're good? Awesome. So let's begin. We're going to talk about the different techniques that I'll be applying for today's lectures across the first, second, and third session.
that Tambutilla Yanga Suga Tirangil Hotel Simiorwa, so the Tampunya Simbaga Caps, Pechitangil Hotel Tablan Kongochigis.
So throughout the first and second session, and perhaps even the third session, we're going to do what we call a pedagogical drawing, which is a drawing of a model that interlinks the information that we are sharing as a class.
That dinner Asuki Pechetangachika and Hotel remote drag at Talenting Asudus Pechetangis, so the Tampola Tangare, Nuala Pechetangare, Chichinas, and Balapechetanga Kores, the Digar system Malaya, Natsugi, Zentanani Trishagi, Neton de Tarwa, Tetan Drevi, and Pizuchi, and Ermula Truba Kigres.
So throughout the lectures today, I'll be asking you to stop, talk about it in a group, and make different models of different topics that we are covering. Okay?
Ta Zenda Trishing Karchigre system, Bala, Zenda Tis Yanko Samsha, Nishetsula, and Tandanga Su Trisha Jung Data Drevi, and Pizumanda Karakarindu, Tichetsuna to the Chis, to the Chinujila, Ermula Parrochi Sagete, Yansi Chigins.
And making models allows us to cognitively think visually about the topics that we are discussing as a group. And that is critical for really linking the information that we are learning from each other.
The Pizu in the Cheba in Baina, Karichigre system Balaya, Susu, Jan Shaju Teta, and Remula Pabacha, Remu Pabateta, Susu Miki Tononachi Yongorwa, the Tonkachuzuwa, Tilatinidete, the Natsugi, Jan Shaju, Nitsa Mampuchka Parolaya, Drewatalaya Pegres.
And the second technique is, of course, utilizing a lab experiment where we are going to apply an experiment, we're going to collect the data, and we're going to apply and assess the results. And we're going to compare different groups to see the variation of the data between the groups.
Ta the Jelang Asukar Sigre system Balaya, and a Taksha Shimji de Pansalaya Davachi Anguresta, the Kasukar Chigresana, Taksha Shinju Denu Jelaya, and a Kasugreta, Trimditelata. Tila Ishipchi, Ishichinu Jelaya, and a Commander Manda Parolaya, and a Koge, a Trimditan Devi Chepakarindu said, the young Sonda Chiata at the Yungus.
All right, so let's begin. So, we're going to talk about plant development and continue with atomic interactions for the first of the three sessions today.
That and Sotan Tampokasim, Bananga session gets Hapertan, and the Tengi Parogin Javach or a digit Tetagins.
Now, again, you're probably wondering, well, plant development and atoms don't really correlate, but the second session, when we talk about photosynthesis, you have to build what you learn on atomic interactions for photosynthesis to make sense, which is why we have to cover atomic interactions.
That's it. By Manachasugi, Tshangi Sapetan, the Tangi Deja said that Joachim do some horse. Imana Sotan Yiba Kasangasu, Ojur Kolashem becomes the Tangi Deja, the Hachikachimuchagores, Nistambalaya, Sota Tampu Caps, and a Ojur Kolashe pickups, Shene Chesungasu, Sotan Tampu Caps, the Tangi Deja Shepate, Alekogi, Minshitan, Manshitan, Juchiri, some grace.
And just like I do normally with my classes, if there are any questions, raise your hand and we can discuss them. We can share different ideas, but I want all of us to really be up to the par on what we are covering together. Okay?
Namjingarang Zenta Trigger the Chiparis, get the Troy Opayam by Labaya Charochi, Serwa, Natsulanja, the Tridichigo Opanachigians. That Hunse Trippy Caps, and it's a Samag Zabzabchi de Dona Yapugan and Yarochi is, Nangasu Degan like Karichigio Pate, and then Yam the Trision Johnson Dugins.
So let's first understand, let's sort of step back and get an assessment of what plant growth and development really mean?
다 당부들라 가지고 저신가 설치해서 growth 설치해서 surface 때 가림베 됐다기인스.
Now, you may think that growth and development are the same thing. Even though they're related, they're actually quite different from each other.
다 설치했던 surface 때 인샬라 두스타바임바나 코란 지바레 삼도 코레스와 임바이나 코란 샤파로라얀 레와다 요레스 임바이나 코란 게 지 마라법지 드로바 임바다 캐퍼 시다 요레스.
Now, growth in general, if we sort of simplify the term, essentially is a reversible increase in the number of cells. And here is my question. What process does the organism go through for it to grow? And we have learned this, we reviewed it yesterday, and we learned it last year. What is a process that organisms go through for growth?
다 단다 데카스 담포테 카레스 담발라야 설치세트레스와 설치세트 네코 칼스그레타 징위나 드코레 야나 왕사 드카사 왕부이나 드고도 야마나 가수 타퐁게 카탕데 네 시도지 메비가네 만트 페와틴달라 고고도와 라 데텔라 가수 설치스그요레스 다 딘데세 바임바네 계산당 룽어마다 가수 따요레스다 설치 누구레세 설치 디치비 값을 저름 가리친 누구요레스.
And that is an open question. So who can tell me what process organisms go through for growth?
징위지 설치지구레스 담발라 띵나나 저름 가린 누구요레스.
Mitosis. Excellent. Very good. So it is mitosis, right? Now, that is actually different from development. Development is a physiological change in the function of how that organism changes throughout its life cycle.
Tande kaso surface te kareses tambalaya tis mandores surface te tis mandores ta kogi cheshoke kholu chigorwa cheshoke kholu tignalaya njwa karendu go yopal hopartane jelly gitene njwa karendu go yopatires.
So in essence, as a plant grows, it goes through different developmental stages throughout its life.
Can you repeat that again?
So as the plant grows, right, as we see in this particular cycle, the plant is growing, but it's being complemented by different developmental stages throughout its life cycle.
Tatanda jingwichi 설치지비 값이 임바나 그런 거다 jingwichi 설치지비 값이 아니 콜라야 아니 철폐기 콜루지거와 지름되기 콜라야 람덕지 있대 냠냠 들고요레스.
So for example, this cycle right here is looking at growth or development. Which of the two are we looking at in this particular cycle?
tatanda dela taba 임바나 가수가 설치라 따기도에 영나 철폐리도스. 그래. 설치를
Is it
생겼어요.
growth or development?
growth를 생냐.
Raise your hand if you think it's growth.
growth를 생이 라바야장으로 제사 설치를 생냐.
Raise your hand if you think it's development.
development를 생겨서 라바야장으로 제사.
So it's about 50-50, right?
같은
I would argue, yes, it's essentially looking at both. But I would argue, because we're going through different stages, right? Sprouting, seedling, vegetative, reproduction, right? Senescence, when it's essentially withering and dying. That is looking at the stages of development.
tatanda 제주 자상가 생각지라고 자고도 usroa 지진나도 순군요보로 지어지레스 임바이나 단다 델라야 지가 너무 내가 때야 델라가 수다 바임바나 다 자상치기 지 갈수 그랬다 세계나네 스고라다 고 갈수 지니기 요바티기 콜루그나네 름바칭 되시 자고도 왔으로a 름바 만도만도시 되시 자고도 왔스 틴 스tambalaya tam보텔라야 고 ता sprouting system balaya ngasuge tirwata koge dela chiwa thambala do ti kapsim bana ngasu de biwa tirwa ti jelaya seedling koge sam memba ne ti jelaya nguse te chirwata ti jelaya koge chepe shebata thamatela nyamgutin juba ti ne tande yong do wa ne stambalaya khorange segena ne segena lan duage ne ramba khasin desi jagu do as tin stambalaya de kapse ngasu phajitela chapel setires development race.
Now you could say, well, how does this cycle relate to growth? And it does relate to growth. From sprouting to seedling to becoming vegetative and reproductive, it does require growth and mitosis for the plant to get larger, to spend energy on its size. But what you can also see is that from reproduction to senescence, there is no growth, but the development changes.
다단다 두스로아 참을 타바임바나 가수게 다 설패공우라 타고요 레스로아 거기 세계 남겨름바 만나마라 진루고요와 임바이나 양스타 름바 데데데네 조아 두코스 추아 톰바톤한테 사우 멤버기 티커들야 루알라 소바디가 데데데 가수 임바이나 가수라 카리루고요 레세나 설치 두고요 레스로아 설치 두고요 바디코 다 차풍라 소바만트 페와라 소바 용고요와 레마레 디난싱게 델라야 अतः चेपे तां दियों पे कब्ज़े में ना गाचुला चर्ची के नेतां ते उनको रेस्ता रे के सालाया अने तंदा के चर्पन ड्रुगु यो पते लाया अने चर्ची को ngū गाने न्याम ते ड्रुगु यो रेस.
So in your experiment that we did yesterday, right? You may see sprouting, perhaps, although I don't know if we have enough time, maybe not. We may see a bit of seedlings, but we'll definitely see some growth in the first stages of the development of the plants that we chose.
다 계산 체수게 그 사본 가시치 주샤요라 사본 주베데 값을 지지바 임바나 단단가 수거든 이마 가시데가 랑라야 니 당보들 스프라우팅 스그로 치와 톰바탄한테 가수게 사원 멤버게 내다시 또가 데데 지지나 당보데 통녀데 지지나 통도 코레스와 니 값이 임바나 가수게 살페게 럼바 만도 통고요레스.
Good. Need to point this to my computer, right? And of course, as we get germination, maturation, and flower and senescence, these are all the different stages, the different changes of development. Some may be related to growth, but not all of them.
다다다 데카세 바나가 수게 사원이 니금 비와탄 타니 차치치 바르와 고데용고도 되게 젤라야 아니 멤바티로와 maturation 되게 젤라야 메토제 바타 되게 타마텔레 મં구요레스 데 참마카레세나 철폐게 럼바 차고요레스와 철폐게 럼바 참마 철폐게 럼바 차고요레스 아니 카시지 임바나야 고 가수고 철치기 우라야 요레스 네 참마 철치 우마레스.
Good. Now, how does a plant grow? And this becomes really, really important. There are two main distinctions of growth in plants.
다 지상라 임바나 고 철치 간다로 치그레스와 데이터 임바나 철치 제탕라야 리치와니요레스.
The first type of growth is called primary growth.
다 그거 리글라야 담보텔라야 가서 그 토미게 철치세 그레스.
Where germination originates in the soil, you have the apical meristem essentially extending in an upward movement.
시가레스 스tambalaya 땅가 주스한테 찍바임바나 거기 제목은 비니투아니 임바나 고 샤자 야라지아지거와 어디레스.
So as you could imagine, primary growth, the plant is growing lengthwise, upwards. Okay? It's getting longer.
다 토미기 철치들라 따바임바나 저한테 야르지그레스와 야르지스tambalaya 아니 거기 런세테 런드루고레스 야르지제.
The second type of growth, secondary growth, is essentially growing on a girth or widthwise. It's essentially growing upwards, not long, but widthwise.
다 버마기 철치스고도 secondary growth. 버마기 철치테 카레스 스tambalaya 야르지고 마르스. And
what happens here during secondary growth, we get the origin of the vascular tissue, which is what allows the movement of water and sugars and all the proteins that sustain the life of the plant.
vascular tissue system kugi jongta ge trapcha tigna la ya ane kugu chuta jula sopaya man reni gi gorwa ti parchichi chegres.
So if you have a plant, you could very easily estimate, calculate, well, how much it has been growing from year 1, year 2, year 3. All you have to do is assess the length of the plant, but also you can also calculate the secondary growth by measuring the width of the plant.
Ta charala ya tshan chiyo pa imbana tshan te ge lo chedo ta ba imbana te ko lo tampo katsu chi sha nyo katsu chi sha simba katsu chi sha se te ya ta yore ku ga rang sala te ta yore te na shin ku ge bom chala te de ta ya ta yore.
And what you will note is that most plants have a budding system where essentially they will sort of escalate from year 1 to year 2. So you could essentially count the number of budding scales and measure the years in length of age of the plant.
Taya shendachi kariyore se stambala ya tshaman shishla ya bat le stambala tiu taya chigorwa gangla ro tiu taya te ane khoge ta lu dek ko jema phegres tin stambala ya khoge gangla yo pi ge tiu ge tangka la tene deta ya na tukse la tene deta ay shin te ge session te ge ya chi lo se te yam to start kres.
Good?
Yes sir.
Excellent. Now, the direction of the growth, we spoke about primary growth, growing lengthwise. It goes from the apical meristem down to the root. This is a bidirectional growth, so it goes from the apical meristem downwards as well as upwards.
다 당부들에 가서 도매기 설치티콜라 쉐바레스 다 단다 임바나 가서 설치가 가설로 쉐바 임바나 tanga suge zemuge biniti wa imbaina tene dete ta tamputle nga su thomi ge sachite ya chig duarwa ta danda de khasola ta pambana ya chigre mala chigres jisa ge chini nishani ya manisha chigres.
And here's my question. We get cell division going downwards. What will be the result of all the growth on the lower region below the soil? What do we call this part of the plant?
다 트라치오 바이마나 데타 바이마나 가서 데라야 다 타풍게 고 자구일라 텐데 데타 정구여와 데기 차시데 고 마시그레스 고라 다 데기 차시데 마시비게 차시텔라 갈라그레스 root.
The root, excellent. And of course we get mitosis, we get the upwards direction of growth. Now you will note as we get more of the different distinctions of the plant, we go through different, and we'll talk about this, cell differentiation, where cells will actually differ. They will become a different type of cell with a different function for the plant.
Ta tanda dikasa takaranja res mas chibatila ngasuga sawas gres ta tanda dine kasu ngasuge ta ko yara chigi doba imbana roa ta tsande ya chigi doba la tende dete ngasuge ta gantela kare yungores na cell differentiation sagdo atapon changes ati yungores ta shingting kuri chenbu chashin korang ga tapon te kanza chagu chigi ane tapon te zo changes chigi yores ta cheli chenten jurgu yores cheli chenten jurgu sestambalaya ku cheli chirap bajunye ge tapon la dus jurgu yores.
And this is important because the way cells differentiate, they are at the mercy of the different genes and hormones within the plant tissue.
Tako changes chiwi kapsu chikarila rale gres tambala tapon tinana yo pe zeda ane tanda zishan tiganana yo pe zishingi menzila rale gres ko karichila changes chi mechite.
So how do plants differentiate? Well, there's a whole process. Well, cells elongate. They get bigger and bigger and bigger, right? They basically become sort of a really elongated rectangle.
Ta zishinga tapon changes kandas chigres ene tampote tapon nerchi chigres rant dugores rimbu chagores.
Once the cell become larger and they elongate, they develop a vacuole. Who in here, because remember we learned this, maybe not even last year, but the last time I was here. What is a function of a vacuole within a plant cell?
Ta tanda dela ranyote bukton satres, vacuole satres. Nanyalo chichina zedana tripic up sensare, chichi paimbaina ya chegu tripic up sensare, rwa. Ni chesu nani bukton sa, vacuole sa digi chele karimba, rwa. Ni treni US.
Anybody here remember?
Say store the water.
Yes, store water, right?
Or energy.
Yes, basically storing water. So if you break a onion and you look at an onion cell on the microscope, you're going to essentially be looking at a huge vacuole. When you look at a plant cell, the largest organelle is a vacuole, and sometimes it takes the whole cell. The whole vacuole takes up the whole cell shape.
That is chunya sochiares, the chunya sochigoresta, Kitty Chorange, any zongi papachi tapolo the stabaimbana, Ra, Tiganalaya, and the Butonto Patongres, Ra, and it's the Shangi Tiganalaya, Ra, the Shingi Taponganalaya, Butonti Hacha Chambu, Resta, the Taton Chishe or Latabi Caps. koga nane ta wangta chishu ti butong chagres rwa ani cham samlaya thapong ge sacha mang chishi sigura da koga mang chishi ta halam thapong ge sacha mang chishi butong ge khang yo chu ge thapong tina yo res.
So let's go through this again. Cells go through mitosis, they become large, they get the vacuoles, and they specialize, they differentiate. They can become phloem, xylem, or even root hairs, and they have all different functions.
Ta tanda di banarwa ta tamputela ya ngasuge dirwa tamputela ya ko chenjur machiwa tirno chenjur machiwa thaponte tamputela ya rimpucha rampucha nu jela ya ko butong te tribra ane butong te tribala tene de te jela ne ko ta chenjur chi de tang khashi bana ngasu di chagdwa karsa Follum said the Chagduan de Badi, Shalamche, young man banana, Sugita, the root hair system balaya, Saug Pusugum, Sauge Putauti Chagres.
So, for example, root hairs in the root, their main function is to absorb water. And of course, the root itself stores energy, it stores starch.
Ta Sawa Gala Yopig, Pusura Chidwa, take Jelly Suti Carre system balaya. Ane ko zawati julde to chuti yankure dires chuti yankure nanglan drengi che guti chigo chelite chigiyores ne zawa imba na zawa ge lekate kare system balaya nuba nyar sochya dires ta nga so starching wala opinion at nyar sochigres.
And once the cells differentiate into xylem or even phloem, well, the xylem actually loses the vacuole and it becomes this hollow channel, allowing the movement of liquid, such as water and or sugar.
Ta tanda ko chenju chibala tene de tanga su ke shelam tanga shenda film se ti chagi ores rota di chabi kapse tanda shelam di imbaina shelam ti gi thapong nangla yopi ge ane buktong te la du res buktong te la du de te la bala tene de te ko nante khotong chagi ores ta khotong chabi kapse tanda shelam se ta tanda di shelam min kro shelam ta banashe Shindongi ta Jontag Nalaya, Chutan Tida Yama Tansagalam Katichagores.
Good?
This sir.
No questions? Excellent.
Dear Samari Kasini Kudish.
All right, so we learned this last year, right? We had a whole activity on the central dogma. I am not going to spend a lot of time on this. I just want to review the information.
Ta de tawa nyampu de ngasu nanjula ta sarwas ngasu chiso mampu tanchi tawa nyampu ngala ta sayus nis tambal de ngantele chiso mampu tange mes chigo ropes chile ngasu jajontes chichigins.
So you recall, we go from DNA, we change the type of information into RNA. What do we call this process? From DNA to RNA? Transcription, right? And then from RNA to protein, we call it translation. We did a whole activity, right? And of course, from the protein, this becomes an enzyme. It gets expressed into a trait. Any manifestation of that particular protein, the shape, the coloration, anything about that organism.
Ta tamputela DNA ngasu RNA chetela ya ngasu ge tirwa phapshirwa phashuche tene RNA protein la ya phapjuche wala tene de ane ko the Pajuala tended the Tamatilaya and a chenam tingongures, Tarwa, Pajuch the Tiziro, Tizingutila tended the chenam tingongures, the chenam said Chingutigi Chechu Karina la decores, Taku China decore, Chechuana decore, Lasso Paro, the Chechu Karim Batiti, and it Tizila tingongures.
We also recall that transcription occurs inside the nucleus of the cell.
Tandonga Sugi Kari Tensuguresena. The
main product is this immature RNA, which is then going to go out through a channel in the nucleus into the cytosol. It's going to be essentially created by this enzyme called RNA polymerase. It goes out, I can skip this, it goes out of the nucleus into the cytosol, and this is not working very well. And we go into translation, where outside in the cytosol, we get the attachment to a ribosome. And the whole product is this peptide, right? This chain of amino acids, which is essentially a protein.
RNA polymerase tanda tizich garwa tila thu de ta ane ku wola ya ku ge di chag do karsa ku ge yeka do chi chag do arwa ti chanu go jela ya ane ti kawala jagre system bala ya nga su ge ta de ge thapung ge ku ge thasi ge na ne ku kangela jagre sena de ribosome na jagre swa tila je de nu ge jela ya ane ti ge jela ya ane tilan jawala tene de te nga su ge de chi go res tap ti kar sa phab ji te chung go res.
What is important, we call this the primary structure of the peptide. It will be a unique sequence of amino acids, and each unique sequence gives a unique function to each enzyme, each protein.
Ta phaju chi wala tene de te nga su phaju te la ya tzi ge changwa chi tgu duwa tzi ge changwa te la ya nga su kar se gre se na tzi ge changwa ge ta primary se ge duwa te tzi ge changwa na ne ta rma tangpu te res thomi ge tzi changwa te res ta de la tene de te correct go res system bala ya ta nga su la ya kar sa tzi tan drebi ge ta ku chena mando yo ra tucha mando te ge yo res.
What is really important to note is that as genes differentiate, different chromosomes have different types of genes. And all those genes are going to allow for different types of cell differentiation.
Ta tanda nga sugarwa thapon te changer jibi kapse kari ngi gugre sena thapon nang la ya nga sugar rigji mambu yorwa te nga chepon mambu yo chepon nang la ya rigji mambu chi yorwa tela tene de te ku rigji mambu la tene de rigji mambu yo patela tene de te thapon imbana thapon te mambu chi la ya changer chit ku yores.
So for example, we have a whole sequence of DNA, right? It has been opened up by our enzyme. We have different segments, gene A, B, C, D, all those different genes. As we go through the central dogma, right, the product of transcription, we have unique RNAs for each individual gene. As we go into translation, we have different proteins that match each unique RNA.
Ta tanda ga su ge chepong ga la ya rigji ko mando yopta rigji ge korang ge tripcha chong wa re la ya ko mando chira tanda de la ya A B C D ta sha yo wa de la tende de te de te phapsi chi wala tende de te nga so RNA ba na ya A B C D mando chung ro was de la tende de te ko phapji chi wala tende de te tzia mando chung res.
So this is important because a single sequence with different interlocated genes express different segments of RNA, and they themselves express different proteins. So we have a multitude of functions through those enzymes in a single DNA sequence.
Ta tanda de na le nga chu ge ngi gwa te kare system bala ya DNA ge ro tanda DNA ge de na la ya ku term chik na la ya ku mando do ro nga chu rigji mando do te mando yo patila tene de nga chu RNA mando cha tila tene de te nga chu ge rigji mando cha go ro tila tene de ta khurong ge jelly su ro ta jelly ta che go te mando chigres.
So that's important. If you are a plant, right, and you have different proteins, well, protein A may be for light absorption, protein B for water absorption, protein C and D perhaps for photosynthesis. So different functions for sustaining the life of that plant will be at the mercy and the function of all those different enzymes.
Tarwa tanda de ti ke chambu chago res ta picha shabai na wo tela nga su ga tji A B C D la subal he doga ta de trip cup se chi che bai bai na tji A T O J ta tji B T chu yang ko rin jya chi gorwa tji se ta de de kare se stambala ya ani ko nga su go o jur chia la pheng yo pala so pa ge ne ta mambu yo res ta de ta kare se stambala ya ko ron ge cheli manda chi doga cheli manda te ta ya session te ge chalong la ra le gorwa te ton joa te ya nga su ge enzyme tanda menzi yorwa menzi te ya ko mampuch ke cheli la tane de yores.
So this is why it's so important to understand that the central dogma in a single sequence yields a multitude of proteins, enzymes, for a multitude of functions for living organisms.
Na hata tanda de kapse bana ro nga su ge DNA ki kuga rambaching bana tini nga sula ya RNA ta dilajin de ta protein ta ya menji su da protein de menji ro menji de ko manda chagu ro stati nga su manda chabate ne ko chuni la ke chu ge menji manda mabu chagu res.
We're good?
Yes sir.
No questions? Excellent. All right. So we have plant cell X and plant cell Y, two different plant cells.
Da de la ya nga su ge zisheng ge ta pon years zisheng ge ta pon X ta Y ni cha yes.
And don't worry about what they are, just see them as different cells. Now we have different proteins, A, D, H, and C in plant X, and B, H, C, F, and G on Y.
Tadi ngi go yo mares ko mando re semnan ko res ro ta tambo te X no na kari ores na protein A ta D H ta C ores Y gna la ya B H G F C ores.
What is important to note is that different proteins, different enzymes have different functions, right? So plant X will differentiate because of the different functions of A, D, H, and C, and Y will differentiate differently from X because of the different enzymes with the different functions.
Ta tanda de kapsa karinge gure system balaya zesha ge thapon X na Y gi na la yo pi ge tjite manda ro do tjite manda system bala ku ge menjite manda ro ni system balaya ku na la yo pi ge tjite ta tjite gi manda imba sonsa ku change je ba imba na ani X na Y ni cha change lo ga chi gres.
We're good?
This sir.
Excellent. Excellent. So we can have, for example, just like what we did yesterday with our experiment, we may see a bit of germination, right? When we get the first seedlings in our germination, the first sort of tiny bifoliates are called cotyledons.
Ta kesanga suge tase chivatere tandanga suge chi saum bigi chiga chigora nyugum bigi chiga te ore sta tampo chi be kapsanga su kare chigresana tanda sondap se ti chigres ta ti kalana ko lomani cho chinchin ching go ta yongres.
The tiny little primordial stem is called hypocotyl. This is sort of the origin of the stem of the plant.
Ta tampo kura karsa ta sabun bibi kapswa sabun ane chi nyugum bibi kapsge de la nga su sonta de ka se bana sonda omala sha dwa ta de ti karesena ku chembu chaba bana zishang ge jongta yorwa zawa jongta sama ba jongta la yenjugres.
And the emergence, remember we have outwards growing under the soil, we have the origin of the root system. What is important to note is that because they're going to be growing in different stages of the plant, they may require different genes for different functions.
Ta shendaji kare sistaba nga suge yanko chiate chi marchi go marchi chibate ta ko mambalaya ko ge zepa yana chawalan jira dire sta di kapse chechu karingi go re sistam balaya kolaya karsa cherpe girwa cherpe ge ramba mando dugo yorwa mando dugo yecha ani sheli mando goya ge ani ko thapong gogo yorwa otingi go res.
So for example, the cotyledon and the hypocotyl are above the surface. They are exposed to sunlight, to photons from the sun.
Ta picha shapa imbana nga su sonde ton sonde omate imbana karichi go resena ani ko sage gangla yostambala ko gangla nyo phoko yorwa.
And what they have, the types of cells that they carry, will be exposed to the sun, and they're going to be essentially identified and signaled by the presence of sunlight.
Ta tenala yobate kare sistambala rwa kuti chia ta te ko ge o phoko yo chans gore ta khola yo o phoko yo pang ngin chit kha ge yang ko ge number te ngores.
Similar to the cotyledon, the hypocotyl is also exposed to sunlight, so it will also be exposed and be signaled by the presence of light, photons.
Ta ya ko karsa ko tanda ge sonde omate gangla yorwa gangla yo pati ani ko karsa ta gangla yo pati nyima go phoko yo chon nyima go la ten de ta datin jungo res ta gangla yo pati.
Would you expect, and this is a question, would you expect the root to be equally exposed to light signals, just like the upper two regions? Yes or no?
Ta tuate kare insana ro nga chu ga chawa chia chidwa chawa gi chashite ne gangla sonde ton sonde oma sa te nansing gi ne wo gi dati phokor wes. No.
No. Why not? Because they are not exposed to the sun, they are underground, so they're going to have a different set of signals, right? So now the signaling that we get from the chemistry of the soil, which is in complete darkness. Very different from the upper exposed regions.
다 강라야 코 니마 포스탄발라야 콜라 니마그 웰라 템비기 코 다티 중국어와 임바이나 자월라 중국어 말이스 자월라 마촌 스탄발라야 아니 콜라야 다튼 땅에테 가리 다기 용고레스 스탄발라야 강라 요피기 차시 땅 만다오게 다튼 중국어와 콜라와 다티 가레스 스탄발라야 지기바나 코 자와티 지사게 사게 나라 요파탄와 사나나 사게 트찰라 텐데다 코 다치긴 거와 테나 신게 가서다 오 사고 올라임바나다 나콘로다
Because these two are exposed, they are signaled by the sunlight, and the root is not, they get signals quite differently, and the development as it grows through its life is going to be very different across the three regions.
그런 게다 단조탕다 다 만도 유장스와 아니고 철폐가 럼바til라로 다 철폐가 럼바 망부치거로 됐다 지피 값스 쿠슨 설치 제탄데 만도요 레스.
We're good?
Yes, sir.
Excellent. So what are the main functions? I'm just going to review this. Don't worry too much about the details. I'm just going to give you some review on the main functions of leaves. So leaves, as we all know, absorb what? Sunlight, the photons from the sun. And the whole reasoning to absorb sunlight is because they photosynthesize. And the whole reason for photosynthesis is to create sugar.
다 대가서 루마게 젤리라 타바이마나 루마게 레카카레스 스탄발라야 니마니 용주고 어디 말할라야체 니마고 어디랜데다 다 랭기 준생카리다 그런가 무게도 카레스 스탄발라야 텔라틴데테 오조치에드로다 오조치와 텔라틴데 타마데 카레스 스탄발라야 아니 카라트리아트레스 garmutriatres.
Different properties of the leaf have also to protect it. I mean, we are in India, extremely hot, right? If you do not have the protection, you're going to be dehydrated and you're going to die. So you have to be well protected from water loss and perhaps disease, fungus, or other types of pathogens that could infect the plant.
다 현대지 가리오레스 스탄발라로와 아니 다 단다데 자가타 웬바나 고 차와치사다 디스탄발라야 로마 코랑코랑가요 고 카르치고레스 스탄발라야 지금 나네 추세야도 당 야나코 나찬종하다 가수게 함부다 드니 현다게 nemben 람다요로와 데타게 네팔인 고에게 송주테 지구 레슬루마게.
And beyond protection, we have to also regulate the movement of gas, right? The requirements for photosynthesis as well as water.
다 현다지 루마게 카르치고레스 스탄발라야 아니 카사 가수게 단다 오줄라 쿠아게 아니 오줄라 쿠비게 랑제당 추디엔 코레네 고초당 아텔라야 당지지게레스.
So in the second session for today, after our first break, we'll spend a lot of time talking about the details of gas exchange and the breakdown of water, right? And oxygen, which is one of the requirements for photosynthesis.
단다 소탄 이바가 값이 바나 가수가 러퍼드 단다 디가사 랑주주로 랑제테 ям코레 질렌치아 타한테나네 아니 오르주르 값스 추페주 땅데타 아니 솔롱트야시거로 어디 간다도이메데 타지우그네타기스.
And this actually is quite pertinent to our experiment from yesterday. So you could see how the leaves are going to, basically the stalks, the stems, are going to essentially come out, germinate, and you can see the first few portions of our very young plant.
나한테 다다 계산가수게 뜻이지와 나신데 델라야 카리오레스나 담봉가수 사본이 컴비오바탄테게 아니 토메기름 바값을 루마 간다로 지지고 요메치고로 아서치기름 바데 델라요레스.
So the first step of germination, which you may see perhaps tomorrow or even the day after, in the first steps of our plant growth, we're going to see, sort of uniform, unfoliated leaves, which are not completely developed yet.
다 지치바임바나 가수니마 카시나틀리 계산치수 답샤주 되게나네 가사 고 철폐때 딱딱지 매주 있을 와다 때 철폐 딱딱지 매주 그다가 소임바나 토막이 지금 로마 지거로 와 어디 제주 동그래스
So you will see a very small, sort of simple cotyledon with unfoliated structures.
아하 당복이 가서 거기 선대니치도 와 당복이 로마 선대니치고도 와 내 제주 동그래스
Above this, we have the first foliation with further growth. We may not get enough time to see this because we don't have a lot of time for our experiment, but beyond the first sort of step of early growth, we get the first portion of trifoliation, which is the basic formation of the true leaf structure in a plant.
아하 다 토막이 다 가서 거기 토막에다 로마로 지도가 되고 선대 춘춘 인식도 와 때 진우가 제라야 안에 takasa nga suge ta luma sumchiwa ge yang chi yongre swa ta dikare se nambala ya chishante ge luma chi ge ta thoma ge ramba de chagres ta de chi chi pai bana nga chola ya chiso shi ta mezang rwa ne dan de chisu ge saben zuba de la tene de ta ko kanda chi ge yome ti chi chi na nga chiso shi ta mezang ne thong thong ko mares.
So keep in mind, as we see our trifoliation of growth, what do we call elongation of the plant? Primary growth. When it gets wider and the girth increases, we call it secondary growth.
Ta tanda tika sangi gwa kare system balaya korwata luma sumchiwati ne yang kurisibi kapsa yara chiatela nga sukar lagre thomi ge sarchi dirwa ani ko bomb dwelaya ani parmi ge sarchi saidres.
So in our experiment, as we start getting some data in the next couple of days, measure the primary growth of your plant. How much is it growing elongation-wise, length-wise? Get some measurements, get some data, and compare it with each other.
Tatanda nga su taksichibi kapsa karichiro chi senarwa tatanda chechu ko thomi ge sarchi ta ani barma ge sarchi chigorwa te nima la chejaro chis rangse katsu chinjwa dosha nima chigri chirerwa ne ko ge singate bomthate katsu chinjosha te chejaro chis.
So let's talk for a minute or two. We won't spend too much time on this. What is a function of the root? Now, imagine, and you have to really, visualize this because we rarely get to see a root system. But if we have a tree, the root system may be 5 or 6 times larger than the actual tree itself.
Aha. Ta denga chuge chiga samlu na tale ya chiga chana che gwa tinda res ro samlu na chege chi gwa tinda res karchin la pa bana namjin bana nga chuge chsengi ko ge chawa ga mala chi ro root system te namjin mi ki thonggo ma ra ko thonggo ma res ta chege bana shindong te ge gangala yo ba res ko ta gangala ya ra che ba dele ani ko ge chawa ga mala te da bungas ki tu chiwa yo res.
So as you are walking outside, just like I do every morning, look at those trees. Those roots may extend beyond the wall of the monastery. They're very, very extensive root systems.
Tachisu shopato alandus kurajab ka sembana yola shindong yo tsusta da chichi pa mano ko su shindong derwa tada de la yang su tsukpa dowa chichi pa mano tsukpa dus ko ta chap chambu chap de te ko tsukpa ge chlo la dus tam yus bares.
So the extent is important because it anchors the plant. It gives it stability and a frame. Without it, it would fall and tilt over.
Tate ko ka chambu chapkres ko ga sawa derwa te zude ka res tambalaya zashanti sagnalaya tembu de ge chedres ro ko zawa ja chambu che chabyate ta meba imbana ko lodu go washington de lodu go res.
Beyond anchoring, they also store nutrients. So potatoes, which are part of the root system of a plant, they're full of sugar, which is why we like them so much.
Ta shendachi kar system bala ku sete sonya chigyo reswa ta picha shabai bana nga chu ga sai ga shokota wa sombai bana yang ko chesang na ne ko chawala thoege ko dibuchi chaga arwa ta te na la ne jodi karyo system bala kara sagora ta kala te ngarmuti shu yo chang kara griti ngarmu yo chang ne nga chu de la shu gawu chigyo res.
And of course, beyond storing food, they also absorb water and mineral salts, which are a requirement for photosynthesis. So when you water a plant at your house or in a garden, how do you water it? You don't water the leaves. You water the ground so it can go below into the root system.
Ta shendachi kare system bala ya ani chawate ge ko lekha karichi ge system bala chu te sagna na yo chu ge chuta ani tarkha ge ri chu ro tarkariti yang kore thangi yores ta teta karla gu yores na ojola gu yores ra ni tamalaya ni susugi damra na la ya metodan sida tinda yo pa na ane chulupi kapse chuti lumaga yaliga mares hola malugores saga lupa na ku zawa na ne saga na ne zawa ti chuti ya juj deta kasata metodan sinte sinte la chutu tokuyores.
So moving into the exposed part of the plant, what is a function of the stem? A very important part of the plant.
Aha, ta chenda chi kare system balaya nga chu ge ta chlo la se gor ta phosa ge session ge chashi chi kare system jonta res ta jonta de bana session ge ko chashi ke chambu chi chag yores.
So stems essentially are the framework, the base, the stability and rigidity of the plant. It provides mechanical support for the leaves and its growing bud system.
Aha, ta chi kare system balaya ko ge chiga drum do ti khotro chiga yores ta shanti la ro jonta ti ge ta ti khora drum ti chiwala tene dete ga su ge gu ga la thiu ta pata ya na luma chiwa te la ya ram do chiga yores.
If you were to cut a cross section of a stem, you would see a network of channels, xylem and phloem. They're very important for the movement of water and sugar throughout the plant.
Aha. Ta shenda karla na ke ti charang ge ta shindong de ge jonta da chi to pa im ba na jonta te na la ya ne chorong ge ka sa ta chi ge buka tang khon ge chashi mang pu chi thong ge ta te na la ya nga su ge sherlam la so pa ge chi buka mang pu chi thong ge ta te na la khar chi go res na chuta langzu ta chi ka sa si chu la so pa ya ma chende chi se lang ga tres.
And of course, it is in the stem that we get the emergence of gametes. Even though we are not getting into too much detail with gametes, this is the process for plant reproduction.
Aha, ta ya jonta ga na senda chi kala nga chu ge chepe la ten de ta nga chu ge gamete system bala ya ka sa chepe ge thapon chi yorwa ten jong go yores wa tan de chala ya nga chu ti ko la ya ta shu shu ten de se ja ge me sim ba na la ti jonta la ten jong go res.
And of course, it is in the stem that we can visualize primary and secondary growth, lengthwise and girthwise.
Aha, ta de ge jonta la ten de te nga chu ge chi che sawa sa ko ta ko sawa chi wa te nga chu ge thomi chi che ta de ge jela ya permi chi che sa to ko ya chi ran do ta bom ten do ge ne ta te nga chu jonta la ten ta kres.
And our theme for today, photosynthesis.
Aha, ta shenda chi kare system bala ya o jo res.
So, mechanisms of growth, germination. We get plant or even the fungus. Funguses are not plant, but they grow in a very similar pattern. They emerge from a seed and spores and begin to grow, which is basically what we did for our experiment, right?
Aha, ta tanda de kapse mana ga chu chisar ge le so chisar kana ro chi chi ge me ta ge ta tampo te ba na ni kun bi ba tres ni ko te bi ge yores ta ga chu ge shin da shen da ga chu ham pu im ba na ya ham pu de shin ma res ro Sesama ko ru ko lo chaku yorwa imba na chitsa chitan te chikpan do res ta tampu te la nga chu karsa yamana sabende yamana hampu ge ta khurang ge thiu do sto ka ku puch to ka o to le te ta tampu ya chi res.
Once we get primary and secondary growth, well, what controls it? Hormones. We get plant growth regulators, PRGs, that essentially control the rate on how fast or slow the plant should be growing.
Ta nga chu ge thoma ta parma ge sarchi te chongwe ge jela ya ne chang ge sarchi te la chisar te la ya tanzan chin te kare system bala ko ge menji te chag yo res ta menji te la ya kar se ge system bala ya chang ge sarchi te la ya tanzan chin is ge res ta te kar chi ge system bala ya ko chichon ta ane joksala sopa kanda do chi ga ne sarchi chi ge yo mete chi gu yo mete tanzan chi ge res.
And one of the requirements for growth is energy. How do we get energy? Through photosynthesis, which is this process which utilizes oxygen, water, right? You have this conversion into this molecule of glucose, sugar.
Aha. Ta shenda chi kare ore system bala ya ane ojo te res ro ojo te kapse kar peche tang re system bala ya ane kuge karsa chita ane nyue te peche tang de ta thamatela ya nga khusu ge karsa ta kutsula kuya ro ta charlong la kuya ani nyuba tetungu res.
So before we step into photosynthesis, like I said before, this is a fairly complicated topic. Before we step into photosynthesis, let's talk about what is happening at the atomic level. More precisely, what is happening within a water molecule.
Ta tampo kro desejayos tanda nga su ojur sate ko jerimte nyokta chauchi yores wa tati nyokta chauti ngu machigo ngolaya tampo te nga su ge the trigger balaya karichungo yo metagins the trigger bala karichungo yo metagins se kasu nga su ge tomatela ane karsa chugi didu yo wa tiger balaya kananu chungo yo metagins.
We're good to move on. All right, let's look at the atoms. Let's look at atomic interactions.
The
All right, so here we have, which of you has taken some chemistry and understands essentially what an atom is in life? So when I say atom, you all know what I'm talking about, right? Excellent. That gives us a huge benefit. So we have a whole list of atoms, right? Hydrogen, the most simple atom. You will see that they have, depending on the atomic number on the atom itself, for that particular element, you have different valence shells, right? And they have unique number of electrons.
The chechula tenete kosola ya the valence system balaya trimbig lodo yosaskorada trimbig lodo yosag quorum te yobache tenalaya ya lodo khatam manda yores.
Here we have the most abundant elements in our universe: carbon, nitrogen, oxygen, phosphorus, and sulfur.
Ta ngasu karsa zamlangang la yarwa onjiktan kham la ya hacham bembelulu yosage ti karere system balarwa ani diris carbon che ani nitrogen che ani solon che phosphorus and sulfur ta bembelulu mambu yores.
And you will notice, depending on the element, you have a different number of electrons in the outermost shell. So 4 for carbon, 3 for nitrogen, phosphorus, 2 for sulfur, and 2 for oxygen.
You said 2 or 3?
Yeah, so these are electrons that can potentially be shared within other elements.
For nitrogen and oxygen, you have like 5, 6?
So carbon has 4, which are by themselves, they are not paired, so they can be shared, right? 4, 4, 3 on this line, and 2 on this line.
Carbon layer shidwa, carbon layer shiche, nitrogen layer sumche, oxygen layer sumroa, the code de by the delaya sumche, delaya nidwa, dekalana ko chamatic bagay lodo chigoroa. And
what is important, and we are going to get into some chemistry, what is important is for atoms to become stable, they have to fill their outermost shell.
The trench tembu de la asra, the trench tembu de la, and a coge tamage corum chigoroa, lodo desag tamaga corum tinaya, a lodo tatachi tango yores.
And once they start sharing those electrons, they create a bond and they become really, really stable.
And how many
electrons they share is based on their outermost shell. So for example, carbon has 4. So how many electrons can hydrogen share with other elements? And that's a question.
Loda katso chinyamchu chitkre te kari la ralegre sana ku trembe gi korum nama yope ga loda ga tanga te la ralegre was ta tene ngatsu ga yanglo la ta bain baina 양롱게 로드 가츠치 냠추치 그레스
Hydrogen.
하이드로젠게
How many? One. Excellent. How about oxygen?
옥시젠 가츠치 그레스
Two. Right. So it will depend on how many free electrons can be shared to fill that final valence shell, right?
아하 다 단데데 가서 가츠치 냠추치 그레스 에티 가레스 스탐발라야 코게 트렘비그 로드가 나라야 로드 라마와 가서요 베텔라 라리기요 레스와 따티 라마와티 젠다 냠추치 그요와
And like I said before, the moment that they come up with other elements and they share those electrons, they become really, really stable.
아하 다 따기 셰바나시레스 다 젠다 또 냠트 로드 치 냠추치 비캅스 코 하짱 템부 차기요 레스
So we can start thinking about different biomolecules based on those sharings of electrons.
아하 다 가서 가르타기 인세나로와 단다가 로드티 냠치치 왔다니 트레페게 아니 징이기 디드만도 같이라타인스
So when we connect different elements or different atoms to each other, they are known as a molecule, a multitude of atoms coming together.
아하 다 단다가 속에 로 connected atoms 스탐발라에 드템 파트주치 바이바나 스타 드템 만다 두스 냠트 디올라테랑 가서 카르스그레스나 아니 디두스그레스
If you have atoms of different elements coming together, we have a compound, which is a complex molecule.
아하 다 케테 로야 디드그나라 디드그나라 아니 디드그나라야 드테 만다와투스 디발라테네 트파이마나 텔랑아스윈 디지스그레스
So here we have two hydrogens coming together, one for this one for this one. They come together, they share those electrons, and we form a covalent bond. This is what we call when we start sharing electrons between two different atoms.
아하 따로와 그 디드 카라 드테 니가 바로라 치와다 파조텔라가 수고 양롱 니테도 스코게 로드테니 냠치초라 두스 트바이마나 델라야 냠치 총와스 그레스와 그 드테 니가 바로라야 저와 총데탄 총와 델라야 아수기 카스 그레스나 디두스 그레스
So single hydrogens are somewhat unstable. They come together, they form a hydrogen molecule, they fill their shell, they become very stable.
아하 다 양롱기 드테 레레레레 임바이나 템보요 마레스 임바이나야 코라니 냠튼 디왈라테네데테 냠치 총왈라테네데테 고게 디드트고요레스 라 타트바티 캅스 디디 임비캅스 코아창 템보요레스
Good?
this
Excellent. There are other examples. We can have single bonds between a single hydrogen with an oxygen, single hydrogen with an oxygen, or double bonds between methane or other molecules. So we could have single, we could have double or even triple bonds by the number of electrons that are being shared with different elements.
아하 다다데 카승아 속에 청와라 타바임바나 청와 세데 바운드 세드로 청와라 타바임바나 카레오레스나 청와 직된데요레스 청와 직된 시스템발라야 저저사가 로드 직당 파저사가 로드 직인 뉴스냠트 고게 냠시치거와 어딜라야 청와 장데스 거레트 최당 메탄라 소바르도 네 청와 니데요레스 니데 카세 니리니리 냠시치거레스
So for example, oxygen can share two, right? Hydrogen can share one. So you have one from hydrogen, one from the oxygen, another one from the oxygen with its hydrogen, forming water.
다 나서 파조 땅포텔라 타바임바이나 코 양롱라야 코 지금 마신 냠치치요 말와 지금 코 냠치치가 뭔가라 로드 지금 냠치치거래 솔롱 임바나 로드 니티 냠치치거와 디스탐발라야 단다 솔롱게 도텐테게 테 아네 카사 양롱 그 도텐니라야 두스 냠치치와 텐데데 가수게 데 추기 디디트고요레스
Nitrogen can share 3 electrons. So because it can share 3, we can have 3 single hydrogens coming in with a single bond forming NH3 or ammonia.
하다 나이트로젠 바이마나 나이트로젠게 코 나네 코 로드 숨티 냠치치트고요. 다 숨차 레레라야 아니 양도 레레레레 지와라테네데테 청조 지와라테네데테 스고라다 가솔라야 티 청와 장대로 레레로와 어들레텐데다 가서 아모니아 트고요레스
And carbon can share 4 from our table, right? So it can share electrons with 4 different hydrogen atoms, and we call this methane, a very important greenhouse gas, CH4. 1 carbon, 4 hydrogens.
Ta ta de bana carbon bana yang ko ji ton nyam to chi chi ko wa de la ya carbon gi du ten chi ta ane yang long ge du ten shi du sun jo la ten de methane se CH4 se de to go yores ta de bana nga so ge ka sa de ajangkhon ni ji jo ra greenhouse ajangkhon ni de ge na ne ane ko ge alangzi ke chem bu chi ga yores.
And just like carbon can share 4 electrons, it can also share in a different variation where you have double bonds with 2 electrons being shared at the same time. For example, 1 carbon and 2 oxygen, and on each side they're all sharing not 1 but 2 electrons.
Ta data by banner Pajotela by banner carbon ge korange lodo shi de korange nyamchi chit kyo Tatela ya nyamchi chita ya mando yores ta tampotela ya ko ge yanglong ge de re ta nyamchi chi so ko changwa changden de wa imba na de kap se imba na nyiri cha yores wa ta de kap se imba na carbon chi la ya oxygen ni la ti wa la ten de ko ge lodo nyiri dus nyamchi chi yores.
So that would be called a double bond, and we could even go with a triple bond where nitrogen can share 3 electrons. It can join with another nitrogen, forming a molecular nitrogen and 2 sharing 3 electrons each, called triple bond.
Ta danda ge carbon solo ni den carbon dioxide ti ba na ko karsha kore se na ko parola ye chwa te ni den de do wa ya yen da chwa sam den de yo res tu bisha sha pa na nitrogen ta wen ba na ko nitrogen ge du ten ni nyam ten de wala ten de te nitrogen ge di cha go do wa du chi cha go do wa because ko ge ka sa log de sum du nyam chi de yo res.
So know the distinction between a single bond, 1 electron, double bonds, 2 electrons, or even triple bonds with 3 electrons being shared.
Ta da de kapse wa changwa changden ta ni den ta simden ji bi kapse kare yemen te ko ka sangi wa chi ro chi changwa changden system bala ya lo de re nyam chi sha yo ro ni den system ba ni nyam chi sha yo ro simden system ba sum nyam chi sha yo res.
Okay, so let's talk about electronegativity. Very important. Now, what is the charge of an electron? Is it positive or negative?
Ta nga su ka ta ge mena lok mush sa te la ta ge yes ro ta che su le charge yes ane lo de le cham ba na ane ko ge khurti ka res for away more is negative.
Negative, that's right. Very good. So, we have a scenario where we have 2 hydrogens coming together, equal sharing, right? We do not have a charge. This is a non-polar covalent bond where we have equal sharing between both atoms.
Ta danda de la ta bai na swa ta danda de kap sa nga su ge yang long ge de chen ni nyam de la ten de yang long ge de chi ge ro ta nyam de de pi kap sa ane ko rang ge nyam chu chi ati ane ko cha nyom be ga ne nyam chu chi sha yo res ro cha nyom be ga ne nyam chu chi stam bala ya de la ya ani yo ma res ta ko kasna lok ko khurte yo ma res de dila.
In the case of water, my friends, we have 1 oxygen, 2 hydrogens, but are we sharing those electrons equally? Yes or no?
Ta danda de ka sanga su chu de de la ta ba na cholong ge de ten chi ta yang long ge de ten ni yo wa yo ba kan shi la kho ge de ka sanga su log de ti nyam chi chi ge yo wa ta nyam chi chi ti cha nyom bu gan nyam chi chi ge yo es. No.
No, right? So what is happening is that those electrons are being pulled towards what? The oxygen.
Nyamchi chigiyo maka cha nyombe gane nyamchi chigiyo ma reswa nyamchi cha nyombe gane nyamchi chigi mestambalaya ane ko nyamchi de chigiyo re ina cha nyombe gane nyamchi chigi mestambalaya nyamchi chi shajigo logdoti ane cholong go shoglaya shuchiwa tensha yores.
So instead of a non-polar covalent bond, we have a polar covalent bond with a charge.
Ta phajotela yang asugo yanglong ge didi imbaina khoge ni mer doga ni mejoge 데르도와 냠치 총와르도와 다다다 출라 임바임바나 저 니탄 덴바 차고요레스 니탄 덴바 차고르세나 거기 드드데라야 록 크르톤 덴바 차고요레스
You will notice this little symbol in the oxygen and 2 on the hydrogens. This is known as a partial. You have a partial negative with more electrons towards the oxygen and a partial positive towards the hydrogens that are losing those electrons.
다단다 데라야 체수게 데도아 카사 솔롱당 양롱가라야 딱한데나 만도 통고요레스 딱 만도 통야데 가레스나 데카레스나 데 총고요레스 데게 가서 데라야 초그르비가 로컬스기도 왔다 로컬 차점로와 쿨 차점이 없었던고요. 레스다 그 냠추 지기요레 냠추 지어만나 찬연부기 냠추 지기 매자 같이 지기라야 쿨식 치어요. 바탕 같이 치어요. 봐. There's a question.
Question, yes.
찬연 맞아비 준세 카레스 단다디 로드드서 오스제사 타냐도 니 준세 카레스
What's the reason behind? What's the reason behind the sharing electrons pull
Because
more
oxygen, my friend, oxidizes. Oxidation is essentially the pulling of electrons towards those atoms. So oxygen is actually very toxic under certain conditions. Oxidation is essentially pulling all those electrons towards the oxygen atom.
다 이야카레 시스템 발라야 단당아서 오xygen 티가 가서게 솔롱 뎁주치알라식 첨부요. 투송 뎁주치알라식 첨부요 시스템을 가서 그랬으나 파족 로드티 수족살라식 첨부 탱그레스. It doesn't. It depend on the electrons that have.
It depends on the number of electrons.
Okay.
Oxygen has 4 unequal electrons, making it electronegative.
다 이제는 만나 그거 놓고 무시시키 좋아요 봐야 땡겨요레스로. 콜라야 솔롱인 바나 솔롱가 달라야 가서래 현다야 시어와 시스템발라 그게 쿠쇼라야 코라 다 솔롱라야 시스템발라야 그게 콜라이 무시티오레스 무시티오 스템발로 무시티오 스템발라 수 탱그레스.
눈에 제너라 게다 솔룰라 디 로드 마야디 잖에 아니 하이드로제가 로드도 파 비셔 작기라 줘라 타니르 만로 그기라 줘라 눈에 제나 로드 비셔 작이라
In a general sense, if you have more electrons,
Yes.
that may repel the other electrons. In the case.
Right. But oxygen can have 4, right? Because of its unfilled balance shell that can share 4. If it has 4 more, they're going to be pulled towards the oxygen, right? So atoms that are electronegative will repel each other. But when you have unequal atoms, hydrogen very different than oxygen, right? Oxygen more electronegative is going to be greedy and it's going to pull those electrons towards its nucleus.
다 chirtangi bana gasu ta ko logdo im bana ko mando ka shipa im bana baja tang ras ta tanda de im bana ta ko ge kasa cholong ge thagi rambatina tele ko logdo changyo marwa 처음에 스탄발라야 그거 젠데tin 다 그게 티나 큰거 지그레스 거다 도도 셰파나 그거 단다 그리디스도 와텔라야 다 동감 그 강라요레 수수티 땅부 큰거 지그레스 땅가 속에야 되게 찰라 셰파임바나 거기 체추거는 셰파임바나 양둘라 따바임바나 아니 가서 솔롱데 카리오레스나 콜라 무시 쳐요레스.
So for example, I don't show it here, but if you have 2 oxygens coming together, it would not be electronegative. It would be nonpolar.
다야 델라야 때문에 스케팅 아수고 솔롱이기로 솔롱도 텐냐도 제바임바나 코니통 템바 차그마레스 니 메주 차그레스.
Does that make sense?
됐어.
Excellent. So we can see water has a partial negative towards its oxygen, partial positive towards its hydrogen side.
나하다 단다델라야 가수고 추긴 드드들라다 바임바나 추기 드드들라야 아니 그게 쿨와 쿨티 차점바 가서 로컬 모티 아니 단다 그 솔롱 쇼다 아니 로컬 포기스고라다 차점바데 콜라 쿨 차점바데 콜라 도스나 아니 양롱 촐라요레스.
And we can now understand the whole spectrum of equal sharing and unequal sharing. So we have hydrogen, 2 hydrogens coming together, equal sharing, nonpolar. Methane, carbon with 4 hydrogens, equal sharing, nonpolar. Ammonia, unequal sharing, right? We have a partial negative towards the nitrogen and positive towards the hydrogens. Water, as we just discovered, oxygen is pulling those electrons, negative in the oxygen, positive in the hydrogen. And I'll talk about salt in just 1 second.
Aha. Ta danda dila tabai bana ya ngasuge koge kurti karsa mukurtan pukurti chatsambage yanko chapsumando rest pisha shapai bana ngasuge cholongge maga cholomasta yanglongin didde ta ane methan la chabu yamba bana koge 냠치 지와테 찬용보가는 냠치 지고요 장스 찬용보가는 냠치 지고요 장 콜라야 네요 마레스 네요 마레스 다다다 데컵스 아모니아 타 출라 촌바임바이나 아모니아 컵스 네 나이트로젠 라야 코 무크르 차sam바타 아네 티게 탈라요 페게 아네 양도 뜨줄라야 푸크르 차sam바도가 이스탄발라야 코 네탄 덴바레스 티난지 출라임바이나 아니 하이드로젠 라야 하이드로젠 샬라야 푸크르 차sam바체 아니 솔론데 라임바이나 고고 무크르 차sam바요레스.
So we end up, and this is important, we end up with nonpolar sharing, right? We don't have a charge, they're equally shared across the atoms. They're nonpolar covalent, polar covalent, where there is unequal sharing, partial charges on different parts of the molecule. And in salt and other interactions, is to the extreme where it is not even unequally shared, it's donated to the other atom, right? So now the sodium, Na, does not have a partial charge, it has a full positive charge, it lost the electron, it is no longer shared, and chloride has gained an electron, right? So now it's negative. So this is not a partial charge, but this is a ionic charge, where it has a full positive and a full negative charge.
Na hada pajoga den shambana ngasulaya covalent bond ro covalent bond se na nyamche chawara nyamte peche tang go ro akro nyamche chi de tan tsugo resta pajatelaya te karsa yanglon ta methane bana nyamche chawachi nyamche chawen ba kan silaya ni me chu che ro ani methane ta chuin bana nyamche chawen ba kan silaya chacham ge khriyo sarwa khur chacham yo sar do 냠치 지고요레스 냠치 지탄테 고 파자살라 시추아 저살라 시추아요 자 콜라에 쿨 차점 그 쿨테용 거와 때때 인상아 수고 냠치 총화나네 아니 유입다 니 메바트레스 니 메바타니 유바트레스 다 셴다치 차고레스 다 가수고 차그리테 소디움 클로라이드 디나도라 춤바임바나 다데 냠치 지고요 마레스 베트쿠라 치카나네 라 치칼라야 테바 토고요레스라 치케 로드치 라고요레스 치케 로드치 테바 토고요레스 틴스탄발라야 피샤샤바이나 소디움 그나네 치라샤요레스 그래서 고 로드치 라스탄발라야 코테 로드치 라스탄발라 쿠푸차고와 아니 콜라야 로커 테파치 토고요레스 토발라텐데다 고 쿠르티 도마차고와 디 갑서 가수게 다 아이닉 반 시스템발라 가수게 디 갑서 지두 청와트 차고레스.
So let's say I'm hydrogen, we have another hydrogen, we are equally sharing the electron, hold on to it, right in the middle, right? Non-polar, there is no charge, equal sharing. But let's say Dawa is oxygen, what is he going to do? He's going to pull it, I still have it, right? But he's pulling it away from me. Unequal sharing, right? Polar covalent bond. What will happen in salt? He would take it from me.
Sorry.
He keeps it. I lost it. I'm positive, I lost the electron, he gained the electron, he's negative. That's how you want to visually make sense of equal sharing, polar covalent, polar non-covalent, and ionic. Okay, thank you.
다다다 대체선로와 대 담보들라 가수게 찬염간에 냠치치고레 시스템발라 파랄라 되기도와 로와 디 오텔라의 찬염부기 지금 시스템발라야 라포요포 냠치치고 바kan실라야 치케쇼라식 추아도와 다 저조들라야 가수게 지드고 총알해 시스템발라야 다 파조탈라 요마 솔렌샤요 레스로 시클라발라요 레스 다 디나도 바나 야세수 삼루 타임 바나 양코게 총알데탈라야 lulacherlilayo 레스.
And what makes water so important is because of its partial charge, it's able to interact with anything in nature that has a partial charge. And most things in life have a charge.
다 단다 데캅스가 그 출라 따바임바나 출라야 거기 그 차잠바데 도와라 차잠바기 유장스다 체춘들라텐데 거기 양 그 차잠 유비기 아니 디드 챈달라야 거기 데저치트크요레스 데와치트크요레스 라 다 챈다치가네 따바임바나 랑숑 캄라 유비긴 디드 마치와 아니 그 차잠 유주레스.
So as you could see, these are just different ways of visualizing water, different morphologies of water. We have one oxygen pulling its electrons with its partial negative, hydrogens partially positive, just different ways of representing. We end up with a tripod where you have one oxygen, two hydrogens, and of course, most living things are made up of water.
다 단다데 타바임바나 데카레스나 가서 다 세속나네 치데 간다로 치케 첨부 예매글라 소파기 촌트테레스다 간들라임바나 지 데이터 사마가서 추긴 데데티 고 간다가네 촌마촌라 소파레스와 데임바나 가서게 양롱제 데라야 솔롱제 아니 코 냠치치와 제르와데 젠다 코랑라 요피기 시우티 두스타데 촌샤요 레스타 촌토만다만다만다 레스와 데임바나 양코 Karsalana compass me yuchuno mochilayan kutsambachinoa, kutsantamanda res. That is and then I got that Seshola Shabambana, that Tapon la Shabambana, Tapon ko Tapchagaja Manchi race.
So this tripod morphology with those two hydrogens that are able to bond and share other electrons makes water a universal solvent interactor in life.
That dela yopic could tripchala tilatene de roa. Ani ko karsa tripchatila tripod system balay ko yata karsa gureta nyangla simtong demba sigoro da ko chashi simtong demba dilatene dete ani ko tiginangla ya tanda khaparta yanglo yo balatene de sigoro da dilatene dete ku triptang dilatene dete sigoro da tarwa jigi triptang ko kopan dilatene dete chuse de tanga suge universal segdo wata yongchap ke solvent lana jurji segore ta jenda samana jutsage ko jichi chagor ngibu chagores.
And the evidence for the importance of water is that, for example, bacteria and humans, we are about 70%, 75% made up of water. Simple as that.
Ta chechola kana kechemo meta bana gangatela ya simbuche tene ngatsu milay taw chombai bana re
So very important, tripod morphology, water is polar and it's going to interact with other water molecules and it's going to form what we call a lattice structure.
Tanda de kasu ngi gwate kare system balaya kuge ta karsa tripod system balaya kola ya compassum de ngundo chitego de kare yemenda tinala ko ani ko karsa chatsum polar segdoata polar system ba ni yopatires ta ni yopatila tine data yanko chugi de gent nyamte yanko ko ditkiyores dela tine data lattice segdoata ta gasu tamik ngudo uchi chagdoa ramadi ditkiyores.
And all you have to do is draw it out. If you have a water molecule, how can you attach one water molecule to another water molecule? Well, you're going to attach the oxygen to the other hydrogens of the other water molecules, and you end up with this really unique universal structure of water.
That this tonight is chicken did the chicken did the gendala condas kodawa jogre system balaya and ko solon de chicken did the genda yanlon to yam to dus and karsa kutubalatin de ta mood the chitugres.
So on share electrons and the share electrons result in a bent tripod, a very unique property only seen in water, making it the universal solvent for life.
Tanda de Kungo de Compassum do Chuchi Trashor, take a relatin gore system balaya, and Karsa Solon de la Yopigi, Korange, Lord Kasichinam Chichi, Kasichinam Chichi Marwa, Tilatin de Tanta Kugut. Kuyig the Skurataku, band shape molecule segduatis, Kukuyo pigi, the digitagures, Kukuyo pig in the Duchila Tribal Atenedete, and Nico Ta Karsagurita, Yongcheg, Yongchapgi, and Judzi Segurataco, Chalatidana Jutkiagi, and Mutil Chagures.
So we have resulting a polar molecule, very important, and we're going to take a break for our tea break, and when we come back, we're going to start with our drawings.
아하. 다 단다 데카셈 바나가 수 추근 드드데 네탄 덴바리도 와 어떤 게 바치로 체스 네 단다 소체 퍼센체 소체 퍼센 사바인 바나 내가 수고 드튼 거 문제이스
Before we go, are there any questions regarding the material that we just reviewed?
아하 단다 셰피 콜라 좋아요 사 소체 퍼센 마탄 거나 좋아요 사 아하 아 So my question is mostly the root system. It appears to be like whitish.
Whitish.
Yes, not in a dark or greenish. What is the reason?
Well, that will depend on the species. Some plants have white roots, some have dark or brown roots. It will depend on the species. What is unique is that a lot of roots look white, and that is due to the presence or lack of coloration underground.
Lack of?
Coloration. There is no color underground. There is no light. There is no presence of exposure to photons. So we lose coloration underground.
Taya ko karsa niru mandala rally grace yang kashi imbaina karpoche roa. Kashi imbaina yang si nakpotang kashina muputinda yores roa. Ta sigi chani shepa imbaina ta ko ta kapundo chi nangyo roa. Ta te kari system balaya ta discoloration system balaya nga sugun do ti mapper jwa sugora ta do mapper jwa ge desigores kachila pa imbaina do yopate nga su te new popalatin chungyo roa. System balaya sag olaya nakon on ikuti ka wo popagala sopa ge chente tege meza ani karpongongo yores.
Some species have color, some don't.
Aha. Ta khashila ya bana sawala ya chontra yores. Khashila yo mares. Aha. Discoloration the discoloration system balaya wo karsan do maprenjwa. Koron gon doksgorwa. Aha. Yeah,
Thank you. Let's take a quick tea break. We'll come back at 11. We'll continue. Thank you.